Today's speaker is Jeff Standish, who's here on the invitation of the graduate student organization. And I see that there are lots of graduate students in the house, so thank you for coming out. So Dr. Standish is an expert in fungicide resistance, and we are so happy that he could take time to come and see us today. So Dr. Standish did his PhD at the University of Georgia, which is also my alumni, so I mean, go dogs. And before that, he had done his master's at Mississippi State, and he had done his bachelor's at the State University of New York. So currently he's a senior biology project lead at BASF, and he's also currently the chairman for the North America FRAC Committee, which is the Fungicide Resistance Action Committee. So welcome to Georgia.
All right, thank you very much for that introduction. So just to kind of outline where I'm going to go here, I'm going to start out by talking about kind of more of my experiences, what I've done. As I've said, I think twice today, being outside of academia, it's surprisingly hard to put together a research seminar. Much of what I do is under secrecy. So I'm going to talk about what I've done and how I got to where I am now, and then maybe talk about industry as a whole and kind of my perspective on what might be available out there. So I'm originally from upstate New York in Canandaigua. This is one of the Finger Lakes. It's a town just on the north side of that lake. The image in the top corner there is from my grandfather's farmland overlooking one of the lakes. We grew grapes, not so much when I came around, but his brothers, my dad, his brother, they grew grapes and sold them to Welch's for grape juice that you might buy at the store. I also had some animals as well. So after I went to Finger Lakes Community College and then SUNY Cobleskill, so State University of New York at Cobleskill, I worked for a few years before going to graduate school. The first of my experiences was an internship with Walt Disney World. So if you've ever been to Epcot, there is a greenhouse facility there called The Land. It's a pavilion. There's a boat ride that goes through it, which a lot of people take on hot days to cool off. These greenhouses are all hydroponically run, and so I managed one of those greenhouses. A lot of the produce that we made or we produced went to the animals in Animal Kingdom. So this is a really unique experience, and it's one that I thought was really cool coming out of my undergrad. This actually also taught me how to be somewhat professional because I was an undergrad, and then I went into a very large corporation where the point was development, so professional development. From there, I started working for Conrad Fafford. This is a soil medium company. They made all the potting mix materials that went into most universities had contracts with Fafford at one time or another for greenhouse soil products. At the time, Fafford was owned by Syngenta, and I was working in Central Florida at these, I guess, putting out, if you want to call them on-farm trials. They were with the greenhouse facilities across the state. And I was working with one of the Syngenta scientists who decided that she wanted to offer me a position. So I ended up taking a job with her. About a week later, or 2 weeks later, Syngenta announced publicly that they were divesting in Conrad Fafford, going to Sungrow. So I sort of jumped companies as they were cutting us loose. At Syngenta, I was exposed to plant pathology. I had taken a course in my undergrad, but I wasn't that impressed, to be perfectly honest with you. I got an A, but I don't know. Instead, decided to go to Mississippi State, though, after my experiences there and do some fungicide and fungal pathology. So I looked at Cercospora saginus, so that is frog eye leaf spot, and I looked at that across the state of Mississippi. And I'll get into that in a little more detail. After that, I went to the University of Georgia, where I worked on fungicide-resistant Venturi effusa. This is pecan scab. Those of you that know apple scab, it's a close relative. And so this was a really cool system to work in because I'd never worked on a large tree and they're 100 feet tall. After that, I postdoced at NC State and I did genomics, metabolomics, and some translational plant pathology. So I have a pretty wide swath here to get through. So just to go into detail on these, I managed the hydroponic greenhouses at Disney and gave 1 hour long walking tours. That was my job. At Fafford, I was evaluating those different soil mixtures, and I was also presenting growers with observations and data. And I would say that that job with Fafford is why I like research so much. That's how I got into it. With Syngenta, I was assisting the lawn and garden scientists with fungicide and insecticide trials. By the end, I was doing them myself, from plot setup, mixing treatments, applying, taking ratings, data entry. Really set me up to move into the next phase. But I also, with no experience in turfgrass, implemented or established a warm season turfgrass program. We planted it all, basically. I sodded it. So that brings me to my master's. So I took that turfgrass pathology research experience, and again, I would never call myself a turfgrass pathologist, but that was the experience that got me into pathology. I went and worked for Maria Tomaso Peterson at Mississippi State. She's a turf pathologist there. She had some soybean funding for one reason or another, and so I was the student that got on that project. I was managing turfgrass trials. That was kind of how I made my assistantship for a time, but I was also traveling around the state, going to all the sentinel plots in Mississippi, looking for soybean rust. We had taproot decline, frog eye, and then some other diseases. But my thesis project was to look at fungicide resistance in this pathogen to the QOIs. So the quinonoxide inhibitors, or FRAC11s. And so those are typically caused by 3 mutations, F129L, or substitutions, G137R, and G143A. The latter is undoubtedly the most important. This basically makes the fungicide water when it's applied to the pathogen. And before I get going on this, this was over 10 years ago. This was a new issue when I was on this, and it is not anymore. We know it's out there all over the place. So across the state of Mississippi, I collected over 600 isolates, and that's from 73 counties. And so the counties on the left that are in green are where I went and sampled. This was in 2013 and 2014, and I was able to, with no PCR experience, develop this PCR RFLP assay to be able to identify whether we had G143A or not. I'd also back check it with everything that didn't. I would sequence just to make sure that I was actually accurate here. But needless to say, we found it in over 93% of our isolates. So in essence, it was widely distributed across the state of Mississippi. And then in the next year or two after that, it was obvious that it was widespread across the country. So after my time in Mississippi, I was still interested in doing research. I never had a question. It was time to go do a PhD. So I went to the University of Georgia, where I worked with Katie Stevenson and Tim Brenneman. And so I worked on pecan. I had some side projects in peanut, which was really cool, but ultimately pecan was my crop for my project. And so I was looking at the mechanisms of resistance and really the epidemiological implications and how we can manage scab caused by Venturi effusa. And I do want to point out, we have this picture of the tree over here. Mature pecan trees are over 100 feet tall. This represents a really big issue when you're trying to manage. And I'm not making a pun, it's a problem. It's really hard to manage pecan trees when they're that big. You have leaf scab, which we have in the middle here. This is your inoculum source. It's overwintering in the wood on the tree. So the new growth coming out is providing your inoculum that gets on the leaves. And then by the end of the year, it's on the nuts that you see here on the right. And so you can manage this in a few different ways. One would be scab resistant cultivars. And that's great. But pecan trees take 8 years to bear. And oftentimes you've lost your varietal resistance before they bear their fruit. So that's not going to work, or at least it's not going to work 100% of the time. You also have cultural practices. This could be hedging, which is a really impressive task. Basically, you're taking a bush hog and you're putting it up on the side of the tree and hedging the tree, which opens it up for airflow. But even that brings about new growth, which is ultra susceptible to scab. So the whole thing is just a really big problem. Then we have chemical control. I put 7 to 15 applications up there. Oftentimes it's over 20 because they're spraying from April when buds break all the way through to when the shell hardens inside the fruit. So that's from April to about October. And that could be every 2 weeks or every time they can finish spraying the entire orchard and begin spraying it again. That's generally the practice. And so we have fungicide resistance to every major class, the demethylation inhibitors, guanidines, methylbenzimidazole carbamate, organotins. At this time, the SDHIs were not really super effective and not marketed in this segment. They are now. And the quinonoxide inhibitors, at that time, there was no QOI resistance observed, despite being applied for almost 20 years. And so the first thing that I did in my PhD was assess the risk of QOI resistance developing, because this didn't make any sense. We've been spraying QOIs in this arena since like 99, and they're sprayed 2 to 3 times a year. We had been looking at in vitro bioassays, but typically with a QOI assay, and those of you that maybe do QOI assays might know this, but if you're looking at it in vitro, there is an alternative oxidase pathway that allows the pathogen to detour the inhibitory effects of the fungicide. You can inhibit that pathway in vitro, but the inhibitors of that pathway are actually also very effective fungicides against this pathogen. So we weren't able to do that. So instead, I started to look, pulling out of my experience in my master's, started to look into how we can basically assess the mechanisms of resistance in this pathogen. And so with the 125 isolates that were collected in a previous project, which was a grower-funded sensitivity program, I basically identified that there was an intron just downstream of position 143. And so this is relevant because if that intron is present, the internal guide sequence covers our substitution position. And what that means is that you can't have a mutation there. Otherwise, the intron would not excise. And if it doesn't excise, you won't have a functioning protein. So those organisms would have a fitness cost and they wouldn't be present. And so that suggested to me that there was a low risk of QOI resistance. Of course, I also sequenced all of those isolates and found a new substitution at position 137. So research continues to roll on. And that meant that I had, you start with a bunch of projects and then you think, this is all I'm going to do. And then you get a new idea and then you have a new project to go forward with. So I had to characterize this. So previously, we had G137R. This is found in Pyronophora triticirripentis. The codons for the glycine, it's a GGG, but in pecan or in Venturia is GGT. So when you have the substitution change in the first position, it goes from GGT to AGT, whereas it's AGG in the other organism there, Pyronophora. And this is relevant because you get a different amino acid, which then has a different protein structure. We did see some reduced sensitivity in that other organism. It was unknown what would happen here. And now because I couldn't do this in vitro, I had to develop a detached leaf assay. So I went ahead and did that. With pecan leaves, it's pretty difficult, I would say. And I spent a lot of time farming trees in the greenhouse for the leaves that I could use in this assay. And then hopefully you can see in the photo on the right at a 200x magnification, we have our spores, we have our germ tube, and then we have one without a germ tube. And so that was how we were assessing relative germination. And so for the wild type, our median EC50 was 0.002 microgram per milliliter. And when we look at our G137S, we have a median of 0.018, so almost 10 times less sensitive. Now, these did differ significantly. I won't say that this is a full-on resistance issue, but it does indicate that there's the potential for maybe a quantitative shift. And since I left Georgia, the students that followed me have found this at greater frequency over time. So it is at least one reason not to overspray a fungicide. So I mentioned a fungicide sensitivity monitoring program earlier. This was grower funded where basically, the producers would go out into the orchard because scab is such a big problem for them. They would go out into the orchard, collect leaf samples, send them into us. We would screen them for fungicides and within 3 to 5 days, give them a result back and hope that that would allow them to kind of guide their next application. So if fungicide X wasn't working very well, they would avoid it in their next app. But we really didn't know a couple of things about those results. Number one, did this actually relate to efficacy? I didn't know, which seems like a silly thing to ask in your project. But so I conducted a field trial at the UGA Ponder Farm over 2 years to determine this relationship. My thought was that as you have a less sensitive population, you would have less fungicide efficacy. And so I really abused these products on the right here. We had 10 consecutive applications of the different products on the same trees over 2 years. And I collected leaf samples twice per year at the beginning, well, basically in June and then in September. And so just looking over these 2 years, this is leaf scab incidence. All of our fungicide treatments reduced incidence in both years, except for tebuconazole in 2017. The DMIs have long been the backbone of systemic fungicides in pecan. The combination of TPTH, which is fetin hydroxide, and is a contact fungicide that is more of our, maintenance spray, where you can't put out your expensive product, so you're putting out a cheaper one. That combination provided some, I don't want to say synergistic, but it provided a greater efficacy than either fungicide did alone. So then sensitivity was interesting. For TPTH on the left, it fluctuated quite a bit. These are relative germination values. So as they go up, they're less sensitive. So 4 times less sensitive going from that 11 to 41, that's a significant shift. And then 37% over winter. So that was really interesting. So over the winter between my 2 seasons, we basically saw a significant drop or to a more sensitive population. Looking at tebuconazole, it fluctuated a lot, but it kind of fit to the same pattern where we lost sensitivity or gained sensitivity during the year. I don't have a good answer for that one, but over the winter it went up. And so when I tried to link these two, I was thinking, well, I've spent all this time, maybe I have something here. No, there was no consistent quantitative relationship between these two. And honestly, I think a lot of that is because I'm 5'9", 5'10". I can reach about 6 feet. That's the bottom of the tree. The tree has 94 more feet to go. So I think we probably missed quite a bit because we were using air blast sprayers in this case. It was a modified one, but I was still quite a bit of the tree to sample there. Now, with that being said, when tebuconazole was ineffective and our growth values were between 35 and 69, well, that's not predictive, like quantitatively. That does indicate in a way that at that particular dose rate, we probably can expect not to see efficacy. The 2 products together provided greater efficacy than either did alone. And then fentanyl hydroxide or TPTH, the efficacy was reduced, but we saw a lot of fluctuation that was really not captured by any factor that I could see until later when I started looking into it in the lab, kind of as just a little one-off experiment. But I figured out that actually our more insensitive isolates on non-amended medium, we're not very good at germinating. Their Canidia were not. So I call that a fitness penalty. And then over here on the right, we have stability. I didn't really label that appropriately, but basically I made transfers every 2 weeks. It's a slow growing pathogen. Every 2 weeks for 5 or 10 weeks. And then I would test it before and after those transfers, and we see significant drops in our sensitivity. So my takeaway on this one was that if we move away from using fentanyl hydroxide for, say, a year, we might be able to come back to it with improved efficacy. Back to that sensitivity monitoring program, I was a little concerned because I don't know that the growers, where they're taking their samples, are they stepping out of their truck and pulling them all off the same tree? They're giving us 45 leaf samples, but I didn't really know where they were coming from. And then when I first went to an orchard, my advisor and I went out and we were looking at all the scab because it's really impressive. You see so much of it. But we went and saw a hot spot, what I would call a hot spot, where basically it was just the worst in the entire field. But that's not really representative of what we were looking at in the entire orchard. So we decided that it was good to quantify this. So this is a roughly 20-hectare orchard, just to give you an idea of scale. I placed a grid over there so we could see what I was sampling, but basically I divided it up into groups of 15. Those would be quadrats. And then I sampled those quadrats and tested them for fungicide sensitivity. And then so in 20, these are the significant ones that I found. So I had 3 different fungicides in 3 different years. In 3 of 9 instances, we saw, this is all the fungicides. So we see significant differences for all 3 of these. An increase, then a significant decrease for tin or super tin, fentanyl hydroxide. For propiconazole, we see a significant increase in relative growth and then a numerical increase. And then we see it kind of fluctuating with thiophanate methyl, which is a more qualitative resistance profile. So that's our temporal variation. When we get to the spatial variation, here is where I have 3 of 9 where we had significant spatial autocorrelation. And now this is hard to interpret because honestly, the resolution's not great. When you start a study, sometimes you learn things along the way. But in this case, what this means is that like individuals are closer together. And so we end up with basically areas of the field that are more insensitive than others. And so what that tells me is that, yeah, we probably need to spread out. So just to give my PhD research, the implications for scab management, lower risk of QOI resistance in this pathogen due to an intron, that led to finding a new substitution, which may still also lead to reduced sensitivity. Tebuconazole was ineffective in the field when relative growth at 1 part per million was between 35 and 69%. Limiting applications of fentanyl hydroxide now may allow for improved efficacy later, and fungicide sensitivity may vary spatially. So it's important to collect representative samples for the orchard or the area of interest. So I came to the end of my PhD and I thought, well, now what? So I had a field-based training with some lab-based application. You know, I had learned, I did a lot of PCR and DNA extractions, but I ultimately spent more time in the field. So I was leaning more applied. I was interested in translational plant pathology. So I was experienced at writing, reporting, and presenting my results, but I didn't have a lot of grant writing experience. And I've mentioned this, I think, to the students earlier and others. I was really good at spending the money, but I had never brought in the money. So I thought, well, maybe that's somewhere to go. I was interested in research positions. That really is what I liked. Research extension faculty roles, R&D scientists, that would have been in line with what I was kind of looking for with Syngenta. And I had a few opportunities for training positions, so postdoc or industry development programs. I ended up with 2 postdocs that I was looking at, and I chose one of them, and that was with North Carolina State. So I joined the vegetable pathology program. This is led by Dr. Lena Casada. So new crops that I'd never worked on, mainly cucurbits. We definitely do a lot of sweet potato work. Sweet potato is a big crop in North Carolina. In this particular case, I was drawn to the program because I was interested in the translational research. I wanted that training. I also wanted to gain the relevant skills in budgeting, grant writing, project management. These are all things that I knew I was lacking. I also wanted to continue my applied research. So I wanted to be tied to the growers, and it worked out here because there was an extension lab. And so my 2 major projects were surrounding developing highly specific diagnostic markers for downy mildew detection and characterizing mycotoxin and phytotoxin production in Fusarium infected sweet potatoes. And so just to start, the project that I began when I got there was to look into molecular diagnostics of basal downy mildew. So I had said earlier that when I originally signed on, I thought maybe I'll get cucurbit. That's the one that really matters. And then someone else got that. I said, okay, well, hops is another option. Maybe I'll get the hops down. No, I had the choice of basil and impatiens. So I picked basil because I could grow it easily, and it ended up working out really well for me. I had, at this point, 0 genomic experience, none. But I wanted to learn it. So I did. I identified genomic markers for Peronospora bovarii. We had an Illumina draft genome assembly already and some RNA-seq data. So I mapped the RNA-seq data to the genome. There were almost 19,000 predicted genes in that genome. There were 4,400 that were being used in the RNA-seq data. And I was able to basically run this pipeline where we have 4,400 all the way down to 114. And so up until the 1492, right here, that was all on a computer. And then I went into the lab, started looking at marker sizes, multiple copies, and did my PCRs running a gel to identify the marker. And so once I had screened those 114 candidates against other OMI seeds and a very large, diverse, across countries, population of Peronospora, I was able to validate the specificity and identify 2 of them. So I had a marker. So then I developed a way to use it. So I took those 2 markers and developed probe-based real-time PCR assays for detection in seed lots. And so these markers, I was able to pick out as low as 500 phenograms of genomic DNA, which is very, very sensitive. That is equal to about 10 sporangia in a seed sample. And I'm not saying that this is seed-borne by this. I collected seeds off the plants that were infected, and the spores were there. So I also then was working on mycotoxins and phytotoxins in Fusarium infected sweet potatoes. And this was funded by a NIFA fellowship. So I was able to pick up my grant writing experience to get that fellowship. And then I had all the budgeting and everything that was all exciting about that to learn those skills. So Fusarium in sweet potato causes Fusarium root rot. This is a storage disease, which also was something I wanted to get into just to try something new. So this is primarily caused by Fusarium solani. Causes circular lesions. They cause these dark concentric brown rings. It progresses to the center of the root. Typically, these occur after wounding. Roots are wounded during harvest because they're harvested with machetes. It is a very labor-intensive process. I've done it a lot. You get in there, you're basically deadlifting in sand, and you're using machetes to cut the roots off. It's very intense. But it's not uncommon to wound those sweet potatoes. Also, when they go into the packing houses, they can be as well. So once it occurs, it progresses during storage, and then it can also progress during shipping. And this is something it's exported out of North Carolina in a pretty large amount. Anyway, so several other Fusarium species have been known to infect sweet potatoes, not just Solani. It used to be that Solani was the only one that was causing this, but that's not the case. One that's known to cause it is Fusarium proliferatum. So that is has previously been identified as producing fumonisin in sweet potatoes, and I have the paper table showing here. But when I was doing this work, there was no quantitative information on mycotoxin production in sweet potato, so that was our problem. And so to identify and solve that problem, we developed an LCMS method to identify and quantify fumonisin B1 and B2, among other mycotoxins. I ran a very wide screen, but those were the two that we found. I had 37 isolates representing 8 species. And so we detected the 2 fumonisins, B1 and 2, in both liquid medium and in sweet potato samples for 4 isolates, but all 4 of them were Fusarium proliferatum. So it was always that same isolate or same species. And we're able to kind of track how it developed in sweet potato. It was a little more static. In liquid medium, it got worse and worse and worse and worse as time progressed. There was another part to this study. So in addition to mycotoxins, sweet potatoes, despite being some of the more healthy food that you can eat, they also often have been found to respond to fungal infection by producing biologically active furantoterpenoid compounds. Now, these are typically lung and liver toxic, so this is a real problem. Several pathogens induce the production of a 15-carbon furantoterpenoid, but it was generally at this time thought that only Fusarium solani would then reduce it into the more toxic 9-carbon compound known as ipomenol, two on the middle there. And I think a lot of the research on this was done in the 70s, and nobody knew about other pathogens that came in on this. So anyways, Fusarium solani was thought to be the only one that could reduce it. And at that time, there was no information on the ability of other Fusarium species to do this exact same thing. And of course, there was also no commercially available standards for 15-carbon or 9-carbon furantoterpenoid compounds. This was also during COVID, and I was speaking with a company in Ukraine trying to figure out if they could synthesize it for me, and they couldn't. So what I ended up doing was collaborating with one of the chemists at NC State. So we generated preliminary mass spec data and identified a common precursor ion, which we're calling the furan ring. So it has a mass charge ratio of 95. And so you can see that it is a part of our compounds. So because I didn't have a standard, we were at the very least able to run our analysis for that compound and identify what was out there. And so we then developed another method using LCMS to identify and quantify the production of furantoterpenoids in these various species. And I know this table is kind of a lot, or these graphs, I have 2 here, but the general point is that if you look on the left, the 4-hydroxymyoporone, 15-carbon, is actually present in our wounded control, but it's not present in the ipomenol. Ipomenol is not present in the wounded control, which indicates that wounding actually causes furantoterpenoid production, and that the pathogens are then able to still reduce it. So more than just Fusarium solani. The 15Cs, yeah, consistently observed in the wounded control. That is a problem because wounding happens quite a bit. So to kind of wrap up my postdoctoral research here, fumonisin B1 and B2 were detected in both liquid medium and in the infected sweet potatoes. I identified both the 15-carbon furantoterpenoids, there are 2, in samples of all of my isolates, but the wounding seems to also be cause production of this in the sweet potato itself as a defense mechanism. The 9-carbon furantoterpenoids were found in all but 4. I didn't find all 4 of them, but the most important one was ipomenol, and that was found in every one of them, every species. But was not observed in the wounded control. So it does appear that there is some reducing from the 15 to 9. And again, all of that research had been done in the 1970s. So it was, the people doing it were no longer employed. Now my next step on this was to use a systems biology approach because I had the mass spec data and I basically did the RNA extraction, sent it off for RNA-seq. And that was my last day at NC State because then I left for a job. So this was done by someone else. So anyways, while I was in my postdoc, no postdoc is ever thinking, this is it, I'm gonna stay here. This is what I'm doing for the rest of my life. So I had originally agreed to stay at NC State for 1 year. I said, I will not seek jobs for 1 year because Lena was taking a chance bringing me in. She only had 1 year of funding. She knew that I wanted to write grants. She knew the experiences I wanted. She took a chance. The end of that 1 year was the beginning of COVID, so March 2020. So there really weren't that many jobs available then anyways. On top of that, the NIFA fellowship I was awarded began at that time as well. So I really didn't see any reason to move on. At this time, I was experienced in grant writing. In addition to the NIFA fellowship, we also did some foreign ag service, like the TASK program, which was pretty cool. That was for export market development and maintaining export markets. Sending diseased sweet potatoes that might have mycotoxins, might not, to a port in the Netherlands is a big problem for the US. So we need to know how to manage that. And then also some APHIS work for Downies. But I also picked up budgeting, project management. I had new research skills and understanding